This genetic stuff is addressing two things. One is, how do genes determine, at least in part, what we look like and how we function? The other is, how are genes inherited from parental organisms? So that's part of the review. The next part of the review is genes and alleles. This is the most tricky part in terms of the vocabulary. It's really tricky because even geneticists will often use these terms interchangeably. They're not interchangeable, but we use them interchangeably because by the time you get to the point where you are a geneticist, you always know what you mean. And so does everybody else. We use the terms gene, allele, trait, character, characteristic, sometimes even phenotype. We use all of those somewhat interchangeably. They're not interchangeable. In the context that we're addressing this stuff right now, it's pretty important that we get the difference. A gene and allele are not the same thing. A gene is a type of protein. A gene is a type of protein that's located in a certain spot on every single chromosome that is like of the same type in an organism. So if we have 23 types of chromosomes, every single chromosome number 7 has a gene right here. If we're looking at plants, maybe this particular chromosome has a gene for flower color right here. Flower color is a category. It's a kind of a gene. If I tell you, hey, there's a flower out there and it has a gene for flower color, you can't tell me what color it is. All you know is that there is a gene, a sequence of DNA in that spot that influences flower color. That's what a gene is. When you look at a specific gene on a specific chromosome, you'll see a sequence of DNA. When that DNA gets transcribed and then translated to become a protein, what you'll see is that for any gene, there's multiple proteins that can be made. They're all going to be really similar. They're all going to have the same kind of basic function, play the same basic role in an organism. Every single gene sequence right here on this chromosome in this particular plant influences the flower color. They're not all the same though. Some of them might make white flowers. There's no color at all. The protein is there, it just doesn't do the same thing as if it's the allele for purple flowers. So these are like variations of a gene. And we've talked about this in a bunch of different ways. We've talked about it like recipe books in music. One of the students in here kind of referred to it as like a font, right? Like this might be a set of instructions, like pick up the box that is located right outside the door. That means this says pick up the box that's located right outside the door. Same information, but this one might be in Times New Roman, and this one might be in Calibri. Are they exactly the same? No. Same kind of thing, but not exactly the same. And figuring that out and recognizing that is something that's really important. The way that I think about alleles and genes is I ask myself a question. And the question is, does every organism of that species have whatever it is that we're talking about? If the answer is yes, then it's a gene. So, does every single flower have flower color? It does. They all have flower color. That makes it a gene. Does every single plant have purple flowers? No. Some of them have purple flowers, some of them have white flowers. In some plants, these two alleles would sort of mix and you'd get pink flowers. So if every single organism has whatever category we're talking about, that makes it a gene. If only some organisms of a species have it, that makes it an allele. What makes us human is that we all have the same genes. What makes us different is that we have different alleles for those genes, right? So, like, if we were to take, Angel, for example. Angel has genes for his height, and Angel has genes for his eye color, and Angel has genes for like the relative length of his fingers. So do I. But we're not the same height. We don't have the same eye color, and we don't have the same lengths of fingers because our alleles are different. some of the things that we think about may not be genetically determined at all, right? Like you could ask yourself, like, do we all have hair length? And the answer is yes, even I have hair length. How to know? To what degree is that hair length genetically determined? I mean, there is a component of it. If none of us ever cut our hair, if we ran around like cave people, our hair wouldn't just keep growing and growing and growing. Like it breaks eventually. But people might have different like lengths that it sort of into. But right now, that's not what's going on. Our hair length is entirely environmental. That's not a genetic characteristic. But many things that we see in organisms are determined genetically. And with people, we're predisposed to be able to figure this out because we have so much experience with it now. We know. Like when we look at like the length of Angel's fingers versus the length of my fingers, that's genetic. When you look at the fact that I am 5'8", solid muscle, probably the strongest person in the school, definitely in this classroom. And then we look at Angel and you're like, well, sorry man, that's environmental. My strong muscles aren't genetic. They're from the fact that I spend at least 12 hours a day pumping iron in the gym. So that's how I got to be.
How are you here?
Huh? I'm not. I'm in the gym right now, lifting weights. So there's environmental factors, but in this class, we're gonna stick to the obvious things where we know, well, this is like, you know, like eye color is one of the most common examples, because we all know, like, that's not something we have any control over. I mean, even colored contacts aren't really changing your eye color, it's just putting a piece of plastic over it. All right, so we've talked about alleles. Dominant and recessive alleles come into play because organisms like us that have 2 copies of each chromosome have 2 alleles for every gene. Those alleles might be the same, or they might be different. If they are the same, then whatever the allele, whatever like characteristic that allele encodes is gonna be demonstrated. I have 2 alleles for blue eyes. Every single chromosome in my body has 2 alleles for my eye color, and they're both blue. That means my eyes are blue, right? And if I had 2 brown-colored alleles, then that would be the same allele, and it would just mean that my eyes are brown. But what if you have one of each? Well, sometimes when you have one of each, they both kind of do something. There is mixing that occurs when it comes to alleles. Flower color is a really good example of that. Many flowers really do show shades of color that come from mixing of genes. That theoretical picture we were just looking at is actually information about a real plant that was studied by Mendel, the guy who figured out a lot of this stuff, pea plants. And it turns out that pea plants can be purple or white and sometimes pink. So you can sometimes have this mixing thing that goes on. We're going to stick to the simplest case. And our simplest case is going to be that there's only 2 kinds of alleles, dominant and recessive. It's not true, but that's how we're going to play it. Dominant alleles only need one of them. If you have one of them, they will determine the phenotype, the appearance of the organism. And recessive alleles don't do anything unless there's 2 of them. The real thing that's going on with recessive alleles is they kind of just don't do anything. My eyes aren't blue because I have a gene that makes them blue. My eyes aren't blue because my alleles don't do anything to change the color of my eyes. Everybody's eyes would be blue if we didn't have some proteins that changed their color. Blue is like the baseline color. That's why you might know this, especially if you have like much younger brothers or sisters. Most babies are born with blue eyes. You don't get your eye color for at least a few months. As you get older, your body starts producing the pigments that color your eyes. And so for me, I have 2 proteins that just don't really do anything. They don't change the color of my eyes. If I had 1 allele for brown eyes, that would be enough. Then my eyes would be brown. And that was the case with my parents, who both have brown eyes. They must have had, each of them, 1 blue-eyed allele. Because they each have brown eyes, so they have 1 dominant. I have 2 recessive. So that must mean that each of my parents have one recessive allele, but we'll get to that shortly. So how is it that these alleles kind of get distributed in gametes? That's where the meiosis piece comes in. And this gets tricky because we've got multiple chromosomes. Each chromosome has 2 sister chromatids, and each chromatid has multiple genes on it. So what we're gonna imagine, just for the sake of the most clarity, is that every single chromosome only has 1 gene on it. We're just gonna imagine that it only has 1 gene. So this big chromosome has 1 gene, and we'll just say it's A. And this little chromosome has 1 gene, and we'll just call it B. That's not exactly the case. But genes that are on different chromosomes can line up differently in meiosis. In meiosis, homologous chromosomes line up first and then split apart. So this chromosome right here has the same allele on both of its strands. This chromosome right here has the same allele on both of its strands. If this parent organism has 1 dominant and 1 recessive allele, then when gametes are formed, the dominant alleles are going to go into one daughter cell, the recessive alleles are going to go into another daughter cell at the end of the first stage of meiosis, and then during the second stage, those sister chromatids split apart, and I'm going to get 2 alleles, 2 dominant alleles in these 2 daughter cells, and 2 recessive alleles in these daughter cells. That's happening with these chromosomes as well. So these chromosomes have a gene on them. In this case, this chromosome has 2 dominant alleles. This one has 2 recessive. So in this meiosis 1 daughter cell, there's 2 sister chromatids with the dominant A allele, 2 sister chromatids with the dominant B allele. In this daughter cell of meiosis 1, we have 2 recessive A alleles on sister chromatids and 2 recessive B alleles on sister chromatids, which means the gametes at the end have a dominant A, a dominant B, another dominant A, a dominant B, and a recessive A and a recessive B and a recessive A and a recessive B. The chromosomes sort independently of each other. So if we have a dominant allele lined up on the left for chromosome 1 and a recessive allele on the right for chromosome 1, that does not necessarily mean that the dominant alleles will be down here on chromosome 2. They won't necessarily be lined up with dominant alleles on the left and recessive alleles on the right. In fact, 50% of the time, it'll line up the other way. So half the time, if we had 2 chromosomes, and each of those chromosomes had 1 dominant and 1 recessive allele, they would line up so that the homologous chromosome with dominant alleles were both on one side, and the resulting gametes would either have both dominant alleles for both genes or both recessive alleles for both genes. The other half of the time, they line up the other way, where for our chromosome 1, we have the dominant alleles on the left and the recessive alleles on the right, but for chromosome 2, it's the other way. We have our dominant alleles on the right, the recessive on the left. That means our daughter cells for meiosis 1 are going to have dominant alleles for chromosome 1 and recessive alleles for chromosome 2, or the other way around, recessive alleles for chromosome 1 and dominant alleles for chromosome 2. That makes these gametes different. Every time eggs or sperm are made, it starts with 1 cell with 23 chromosomes lined up randomly. Every time meiosis occurs, you end up with 4 eggs or 4 sperm. This is the possible combinations if this is our starting point. This is the possible combinations if this is our starting point. With 23 chromosomes, there's 2 to the 23rd power ways of arranging them at the start of meiosis 1. which means there's 2 to the 23rd different kinds of eggs or sperm that any one human being can make. That is effectively infinite. That's more than anybody here can ever count. Which means that when humans and other sexually reproducing organisms reproduce, their offspring are always genetically distinct from each other. They never have the same combination of alleles from each parent. All right, so this is where we get into Punnett squares, and what I'm going to do is kind of paint a picture of how to use this. We're going to talk and think about probabilities as well, but I'm going to let the Amoeba sisters do that part for us. So with Punnett squares, we are not looking at individual offspring. This is not one baby pea plant, another baby pea plant, another baby pea plant, and another baby pea plant. What we're looking at here are all the different ways that gametes could combine. If we have 2 parents and we know what alleles they have, we can look at all the different ways that they can combine. So for example, if the male plant has 1 dominant allele and 1 recessive allele for a certain trait, and the female plant has 1 dominant and 1 recessive allele, then there's 4 different possible ways that a paternal gamete, sperm, could match with a maternal gamete, an egg. The dominant allele from the male could match up with the dominant allele from the female. This would be a sperm that happens to have the dominant allele in it and an egg that happens to have the dominant allele in it. When they fuse, the resulting organism would have 2 dominant alleles. The male also might produce a sperm with the recessive allele. If that sperm fuses with an egg from the female that has the dominant allele, then we're going to get an offspring that has 1 of each allele. That will also happen if the sperm has a dominant allele and the egg has a recessive allele. There's 2 ways that these 2 parents can create offspring that have 1 dominant and 1 recessive allele. That's called heterozygous. One way is they get their dominant allele from the male, the recessive allele from the female. The other is they get their dominant allele from the female, their recessive allele from the male. And then if a sperm that has the recessive allele fuses with an egg that has the recessive allele, then the resulting offspring would have 2 recessive alleles. This isn't saying that this happened. These 2 plants may never reproduce. But if they were to reproduce, then randomly, on average, a quarter of all the offspring that they produced would have 2 dominant alleles. Another quarter would have 2 recessive alleles. And half would have 1 of each. A quarter of those potential offspring would get 1 dominant allele from the mother and 1 recessive allele from the father. Another quarter would get a dominant allele from the father and a recessive allele from the mother. So what Punnett squares really show us, they don't show us offspring, they show us probabilities. If we were to take these 2 plants and breed them together 1,000 times, what we would expect is about 1/4 of them would have 2 dominant alleles, about 1/4 of them would have 2 recessive alleles, and 1/2 of them would have 1 dominant and 1 recessive allele. It wouldn't be perfect, but it'd be pretty close, statistically speaking. So if these 2 plants were to have just 4 offspring, like if I were a geneticist and I took sperm from one of these plants and fused it with an egg from the other one and then set it aside to grow, and did the same thing again, did the same thing again, did the same thing again, that does not mean that I'd get 1 of these, 1 of these, and 1 of these, and 1 of these. What that means is those 4, each 4 offspring would have a 25% chance of being homozygous dominant, a 25% chance of being homozygous recessive, and a 50% chance of being heterozygous. The laws of probability say the most likely outcome is not that we get one of these, two of these, and one of these. It's actually not the most likely thing. So it's just slightly more likely to end up with more of these heterozygous ones. So the laws of probability work a little funny, and we're going to play around with them a little bit, but mostly what you'll need to remember is a quarter of potential offspring will end up here, a quarter will end up here, and half will end up in these two combined, okay? All we're looking at is possibilities. These do not represent real offspring. Any one real offspring is only going to be reflective of one of these. As soon as you have that, the others just aren't there anymore. Okay, and then just a quick review. I've used these terms a bunch, and we did talk about them the other day, but we're gonna be using them a lot now, so get used to the idea that when we use the word homozygous, it means both alleles are identical. Homozygous requires you to say either dominant or recessive to give full information. If something has two alleles of the same kind, they might be two dominant alleles or they might be two recessive alleles. So we say something is homozygous dominant or homozygous recessive. Heterozygous, there's only one way to be heterozygous. It's to have one of each. Now, there's two paths to get there, right? But this organism and this organism would be genetically indistinguishable, which means that we just say that it's heterozygous. There's no such thing as heterozygous dominant or heterozygous recessive. So if we were to look, for example, at the earlobe thing, which is largely controlled by one gene with two alleles, if you have two dominant alleles, if you're homozygous dominant, you'll have detached earlobes. If you have one dominant allele, if you're heterozygous, you'll have detached earlobes. And only if you have two recessive alleles will you have attached earlobes. Or with the flower, what we were just looking at, heterozygous and homozygous dominant organisms will look the same. It's only homozygous recessive organisms that will have a different appearance. All right, let's let the Amoeba sisters talk us through a little of this. I got you covered here. This poor computer.
I have a confession to make. For my classroom...
Is this working? Oh, no, hang on. Oh my God, this poor computer. It gets so confused. It wants to do a good job, but it just can't. Here we go. Okay, sorry.
So, I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spent quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene, and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter, or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait not having hair shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase little h. A guinea pig that does have hair can be represented as a big H, or a big H, little h. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H, little h is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype? Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H. We don't really know. We could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. Or percentage, 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. But what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. Now remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, they have hair. So 3 of them here, the 1 big H baby and the 2 big H babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls, or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters. I'm reminded to stay curious.
Okay, what we're gonna do here, this is, all right. I'm gonna very quickly go through an example, Punnett Square, then you all are gonna, we'll have a break, and you all are gonna have some time to practice. This is the simplest kind of Punnett Square to do. Punnett Squares where you are told what the genotype is of each of the parents. Heterozygous means one dominant, one recessive allele. A heterozygote is an individual that is heterozygous. So if we have two heterozygotes, and we're talking about people here, and the gene that we're talking about is toe length, if we have two alleles, one long toe is dominant, the other short toe is recessive, then our male parent is gonna have two possible gamete types. Either the gamete will have this male's dominant long toe allele or recessive short toe allele. The female parent will also have two possible gamete types. Half of her eggs will have a dominant big toe allele, the other half will have a recessive small toe allele. The children of these two individuals are gonna have a 50% chance of getting this allele from the father, 50% chance of getting this allele, same with the two alleles from the mother. If someone gets both big toe alleles, they'll end up homozygous dominant. If they get both little toed alleles, one from each parent, they'll end up homozygous recessive. And if they get one dominant allele and one recessive allele from one dominant allele from one parent, one recessive allele from the other, then they'll be heterozygous. When we write the genotype for heterozygous individuals, we always put the capital letter first. There is no convention about which allele came from which parent. It's either homozygous dominant, homozygous recessive, or if it's heterozygous, we put the dominant capital allele first and the recessive allele second. What would this look like? Well, all three of these possible gamete combinations would result in somebody having a longer big toe. This gamete combination would result in somebody having a shorter big toe. So in terms of the percentages, 25% would be homozygous dominant, 25% would be homozygous recessive, and 50% would be heterozygous. In terms of the appearance, 75% of this couple's kids would have a longer big toe, and 25% would have a shorter big toe. If they only have one kid, it's either going to be a long big toe or a short big toe. If they have 4 kids, each child has a 75% chance of having big toes and a 25% chance of having small toes. This couple could have 2 children or 8 children and have all of them have long toes or all of them have small toes. It could be half and half. The more individuals, the more children they have, the closer it's going to be to this 3-to-1 ratio. If they have 10 kids, chances are pretty good that 3 or so of them will have smaller big toes, but it's not guaranteed. Okay. Somebody have a question back here? Did I answer it already? Okay. So there is an assignment on Canvas. It's about Punnett squares. We're going to start it today. There's some mystery solving that goes on that I haven't showed you yet. So we'll start it today. I want you guys to play around with it. You'll be able to get through at least the first 3 or 4 problems. And then as we start getting into the puzzles, those we'll look at probably on Monday. If you want to take a break before you get started on this, now's a great time. Just be back by about 2:35.